everyone. Thank you for joining today. I see folks are still rolling in, so we'll wait a couple minutes to dive into the content, but thanks for joining us. All right. So thank you for joining us today, everyone. My name is Rachel Gauvin. I'm Head of Human Potential and Program Management here at Nucleus Biologics, and we are thrilled to have you here for our Paths to Media Mastery 2, Part 2 webinar today. So we'll have some great content to share, some great data. I encourage you to use the chat and the Q&A boxes throughout the webinar to send comments and questions. We'll have a great session at the end to address all of those. But without further ado, we can get started. So, Brad.
All right. Hello, everyone. Good morning, good afternoon, I suppose, as well, to many of you. Just jump right in, as Rachel said, to talk first and foremost, before I make the introductions, as to why we're here. We're here to see some really, really compelling data that was produced, using formulations that we, or I should say the scientists, generated live online just a little over a month ago, not too long ago. So that's the exciting piece of this. They were able to, in Path to Media Mastery, use MDAir to generate formulations live, as you guys saw. And we were able to deliver, implement, perform those studies, and now we even have data to show you guys today, which is all a really, really exciting turnaround to be able to do. So I will, if you'll advance to the next slide, Rachel, make the introductions. For those of you who were not able to join, and these are in kind of order of in the speaking order. So we'll start off with Asma Ayari, who is a scientist here at Nucleus Biologics. Saba Ghassemi, who is a research associate professor at UPenn Center for Cellular Immunotherapies. Lori Noffsinger, who is in operations and development at Cognate Bioservices. And I'm kind of new to the screen. My name is Brad Taylor, I'm Vice President of Marketing at Nucleus Biologics. And just for those who are kind of new to the topic, new to maybe to Nucleus, we wanted to do a little bit of a rollback and kind of really talk about some of the things that we find so exciting about custom media, why we think it can be transformative, why we think it's really, really important to our industry as a whole. So let's talk about some of the myths that are floating around out there. Firstly, the specifics of your formulation aren't really important. And I'm actually going to call to Saba to talk a little bit about this, but we've been able to show in publications and in the publication history that even single component changes can really improve, things like transfection efficiency, cytotoxicity, yield, potency, as we'll see in the next slide. This was some fantastic work that was done in conjunction with UPenn, and I think we can advance to the next slide. There we go. In conjunction with UPenn, looking at CAR T cells in a xenograft model of glioblastoma. And Saba, if you want to, since you're the first author and we have the privilege of having you on the call, I would like to see if you want to say a few words to this.
Sure. Hi, everyone. So this is a work that we published last year in collaboration with Nucleus Biologics, and as Brad mentioned, we realized that there was an underappreciated role of components in the media, and how, if you change components, that would enhance and affect cell therapies. So here, for example, we showed that if we change one factor, and then here we replace serum with physiologics, which is basically concentrated growth factors, it enhanced the potency of CAR T cells in xenograft model of neuroblastoma, actually. We had mice and we injected tumor cells into these mice, and we had like two groups that we compared with each other. One group was were CAR T cells that are expanded in media that contains serum, and then one group was cells that are expanded in media contained in physiologics. And we saw that there is a dramatic impact in the efficacy of CAR T cells. Even, you know, we did a stress test. We changed the dose of the T cells, and we saw even that in the lower dose CAR T cells that are expanded in physiologics, they clear the tumors better than, you know, their counter group. So that basically told us that, you know, this is like only one component that we changed for this study, that made a dramatic impact.
All right, thank you, Saba, for walking us through that. That is, you know, an extremely important point as well, especially when you take into account the fact that many media providers out there don't truly let you know what's in their media, right? So we need to really take account as to, you know, what we're adding or depleting and how we're manipulating that media, and that's certainly why, you know, custom media to us is, again, something that's very exciting and can be very, very transformative, because if we can use that media to increase cytotoxicity and potency, then that's certainly a game changer for us. So we can move back to the next slide, Rachel. All right, another myth, researching and configuration is time consuming. Well, this one, you know, I'm gonna give it to you. It can be. If you look at the next slide, Rachel, you'll see that there is just simply a mountain of data out there. This is data taken directly from PubMed. It's the number of T cell publications that are kind of currently available on PubMed, and you can see that number there, which is kind of awe-inspiring. Just for T cells, 512,299 T cell publications historically. This is increasing steadily every year. As we all know, there's a lot of research being put into T cells, you know, CAR T cell therapies, as Saba is working on, and others, you know, is increasing, and is very exciting. So, you know, we anticipate this to only increase. So how do you really quickly dive into those publications and find those components or those pieces of data that give you indications how those components are going to affect the outcome of your study? And when you find a bit of information, how do you really determine, you know, is this really the best information? So in other words, how do you canvas all this data? How do you canvas all this information quickly and in an expedited fashion in order to get you guys to your studies the quickest? Well, the answer is, as we're going to show you guys today, you know, and have shown in the past, you know, MB-Lux was kind of one step in that direction by, you know, shortening the configuration, the ordering time, and the manufacturing time. But MB-Air has really tackled another aspect of the timelines that it takes to produce custom media. If we go to the next slide, Rachel, we can kind of start off with kind of your traditional overview of you know, how custom media was produced, and it is kind of time consuming and a tedious process. It begins with a lot of research, as you see, and with the blue area of that bar, moving into configuration and quoting, which is a manual process, which again can be very time consuming. Manufacturing, because it's typically done on large scale, can take a number of weeks to really produce and deliver. And then of course you have to do your testing. This is typically repeated, right? Because, you know, typically there's a second round where you want to tweak something within that formulation. You want to add or increase or perhaps decrease or change something about that formulation to make it even more efficacious. If you hit next, we will see the comparison to proprietary off the shelf. As the name implies, of course, it's off the shelf. So the manufacturing has actually been done prior to it being put on that shelf. So of course your manufacturing time actually becomes a little bit more of an order and delivery time. Research stays the same, but you can see the time to actually starting to implement and utilize that formulation in your studies is dramatically decreased to somewhere around, I believe, 23 weeks. Now, what we've been able to do at Nucleus Biologics, if you hit next, Rachel, will actually compares as far as the timing to what you see with proprietary off the shelf. We've been able to shrink that ordering time, that customization, that configuration time by using online tools to expedite the process. And now today we're taking it one step further with a combination of MB-Lux and MB-Air that further shrinks the research time taken in order to produce that custom media. At the bottom, we have a complete breakdown of the number of days that are involved, and you can see if we summarize the days, it's only about 144 days, or around 20 weeks, 20.6 weeks, to move from formulation to your final media formulation in the laboratory, compared to traditional custom, which comes in at over a year to see those same results. And then again, with the introduction of MBAir, we're saving somewhere between 2 to 3 weeks off of your current timelines. Let's go on to the next one. All right, I can't afford it. This is another one we like to hit head on. At Nucleus, we can scale with you. We can offer volumes anywhere between 2 liters and 2,000 liters. We have very low minimal order quantities. So like I said, you can order 2 liters, and sometimes it does come with a bit of a hit to the price, especially at the small liters. But once you start to move to volume, once you really start to scale your process and scale your studies, you're really going to realize a lot of cost savings. Actually, we did a case study in the next slide. If you're just looking at kind of a case study with high commercial volume, around 5,000 patients a year, estimating somewhere around 14 liters per patient. If you compare the proprietary off the shelf, and this is just an average taken of the media and all the components that one would need to purchase and add to that media, it's somewhere around $300, $299.50. Custom media formulation at those volumes is only going to be with a similar configuration as what you're seeing with the proprietary. It's only $189. This equates to around 13 million a year, and a delta of 8 million between what you're seeing with the custom media and the proprietary. Not to mention the fact that with custom media, you own your formulation. You only have one vendor to qualify, and you only have one process to validate, versus with proprietary off the shelf, you don't own that formulation. You don't know what's going in it. You don't control the supply. You have multiple vendors, products, and processes to validate and qualify. We'll go to the next slide, please, Rachel. And then finally, I mentioned ownership there. That's a very, very important aspect. Ownership, transparency, and then the consistency of supply. Such a huge issue right now in our industry. If you look at the next slide, COVID-19 has added another level of complexity to the already kind of difficult kind of supply chain and consistency of supply that the cell and gene industry was already seeing. So you're seeing, shortages of supply from, cell culture media to reagents. And you see this, in the news articles, in the headlines, from the scientists to other business journals. And, you have to be cognizant of that. Are you going to be able to source all that material? And if you're sourcing different components to go in your proprietary off the shelf, then all of that is on your plate to do. Versus with our model, you're going to one source for these materials, and we give you a very transparent lead time in order to receive those materials. So I guess to summarize and then to really get to the data, which I know this is what everybody is really here for today, I would challenge that, I can't afford it should become you can't afford not to. So at this point, I'll turn it over to Asma. I believe we're going to start with Asma, and Asma is going to do a little bit of a recap for those who may not have seen the prior webinar, just to kind of give you a feel for what MBAir looks like. So Asma, I'll turn it over to you. I think you're on mute, Asma.
Sorry. Thank you, Brad. Thank you, everyone, for joining us. I will start by a quick recapitulation of the main criteria that we selected through MBAir last webinar. So the cell type I selected to work on was T cell, and then for the CQAs I selected, it was to increase viability, increase proliferation, maintain cytotoxicity, and the components of interest were sodium selenite, human serum albumin, physiologic, and L-alanine, L-glutamine. So we're going through, we're going to MBAir to show you how we did that. So and how fast we can set and develop a media to test. So Rachel, if you can allow me to share my screen, please. Thank you. All right, so here I'm on the platform on NBR, and just a quick reminder, I selected optimizer last time, and then for the supplement, I selected human serum. And then for the cell type, it's T cells. So when I hit T cells here, it shows all the data available on T cells that we have. So if I go next, then here at this level, I'm able to select the CQAs that I'm interested in. So my first CQA that I would like to rank as 1 will be proliferation. And I have the possibility to either select decrease, maintain, or increase. So I'm going to select increase. And then the second CQA for me is viability. So I would like also to increase viability. And then the third CQA is cytotoxicity, which is a CQA that I would like to maintain. So after selecting my CQA, I just go next. And then at this level, I'm able to select the basal media that I would like to use for my media. And I will go with what Nucleus recommends. And then from here, I have all the components that are involved in improving proliferation, improving cytotoxicity, maintaining cytotoxicity, and improving viability. So what I selected last time was sodium selenite. So that's what I wanted to test. And then I didn't select, so for the amino acid and dipeptides, I selected L-alanine, L-glutamine, or what it's known by Glutamax. And for the protein source, I selected human serum albumin, and then physiologics. All right, so then if I hit next here, I will have all the I will have here the cell type that I selected, the CQAs that I selected with the levels that I wanted to reach. And then if I go here, I see I have the details of the formula that I selected here in amino acid, vitamins, inorganic salt, and carbohydrates. And you see here we have also the possibility to show the value in either milligram per liter or in millimolar. So for my experiment that I chose to run last time, you see here I have sodium selenite. So the point was to test sodium selenite. So sodium selenite will be the variable component. So I will have 0 here for this formula. And then I would like to use a little higher level of human serum albumin, and then higher level of physiologic, and then L-alanine, L-glutamine, I will go with a level that is recommended here by NBR, which is 434.4. So this is the first control without selenite. And remember, like if I wanted to test this using any off-the-shelf media, I wouldn't be able to do it because sodium selenite is part of IMDM. So if I wanted to test a media without sodium selenite, it would have been a big hassle to have access to such this kind of media. So the level recommended here by NBR is to give an estimate on what's the level that you would use. So what I wanted to do here is to try different levels. So I have low level, and then medium level, and then a high level. So I will choose like a low level 0.017 milligram, and then I will keep here this one at 20, because this is a component that I don't want it to change in my media. And then I will have this one also at 20, and then this will be the second level, so the medium level of sodium selenite. And here I will have the highest level, which is 1.7 milligram per liter, and then I will have here 20. All right. Okay, so then I will select these 4 formula. So 1, 2, 3, and 4. So here I have all the formula that I selected, and then I will save it to NBLux. And this will take me automatically to NBLux. And from there, I will be able to place my order. So it takes a little bit time to upload all the formula to NBLogs. All right, so here I'm on my account where I have all my formula saved. So if I go on one formula and hit view formula and then load the formula into the configurator, you can see here that I have some boxes to check here. So the first one will allow me to select what type of media I want, if I want it liquid or powder. So as I want it liquid, it will be, okay, I'm not going to change anything here. I don't want it to be cGMP, and I'm not required any tested as it's still on like on a small scale test. And then for the quantity, I don't need 20 liter. I actually need only 2 liter. And I want it in a bottle, and then 1 liter bottle is okay. So then I just want to check and to show you that the changes I made on NBR were translated to NBlog. So as you can see here, the level of L-alanine, L-glutamine was brought here to NBlogs. So this was for the amino acids, and then for the inorganic salts, for example, if I go here and look for sodium selenite, so sodium selenite is over here. And so you see I have the highest level that I selected here. So, so I can confirm here and then place my order. And to place the order is really simple. Just enter the address and all the information and then confirm your order. And then after 2 weeks, I received the media that I'm going to show you the data for which I tested the media. So order successful. So Rachel, I'm gonna give you back the screen. All right, so this is the protocol that I followed for this experiment. So I have the control media, which is optimizer supplemented with 5% human serum. And then I use the NBR suggestion. So I have no selenium, low selenium, medium selenium, and high selenium, with all of them supplemented with 2% physiologic. So on day 0, I activated the cells using Dynabeats at a ratio of 3:1, and then day 3 and day 5, I added fresh media, and then at day 7, I proceed with a cell count and then phenotype. So this is the first set of data. So here I have the IncuCyte capture. The experiment was run on 96 well plate in triplicate. So that's why you see here there are 3 images for every condition. So this is the condition without selenium. This is the condition with low selenium, medium selenium, and high selenium. And this is the positive control. If you can hit next, Rachel, please. So as a reminder, my main CQAs are increased proliferation and increased viability, and also maintained cytotoxicity. So when we run flow cytometry on the on this experiment, the data shows that, as you can see here, we have the cell count, cells per milliliter. So as you can see, we have the T cells are able to proliferate in media 1 that doesn't have selenium, media 2 and media 3, the one that has respectively low and medium selenium. But then when we increase the level of selenium to 1.7 milligram per liter, the cells are not proliferating that much, and we can see this here on the IncuCyte capture. So using NBR media allowed me actually to improve cell proliferation and cell viability. And then when I checked also for the phenotype to identify the expression of CCR7, if you can click next, Rachel, please. So you can see here that compared to the control media, the cells, the expression of CCR7 is higher. Which means that it translates to less exhaustion in these cells. So actually, using this media allowed me to have a quick access to a media that improves proliferation, improves viability, and maintain phenotype and cytotoxicity. So using NBR allowed me to create a custom media without selenium, which wouldn't be possible unless I would go and buy all the product myself and make all my media, like for, I don't know how many components, something around 60 components. And it would definitely take me more than 2 weeks. So using NBR helped me reach my goal, which is increase proliferation rate and increase viability. And it also gave me this piece of information that it's really valuable because it allowed me to identify the level for which selenium is toxic or non-toxic. So now I know that if I don't add selenium, or if I use selenium up to the level of 0.17 milligram per liter, I know that this is not toxic for my cells. And then the level of, if I reach the level of 1.7 milligram per liter, the selenium becomes toxic. And this allows me also to develop a small scale protocol because I was able to place an order for only 2 liters, and it also still gave me the possibility to translate to higher volumes in case I want to scale up and work on bigger scale. So for next steps, I will be testing the impact of other inorganic salts. So I don't know if you remember when I was setting the experiment, under inorganic salts, there was sodium selenite, but there was also lithium chloride. So for next step, I might test the impact of lithium chloride on cell activation and cell proliferation, which will allow me actually to identify which are the critical component for each CQA, and then help me optimize my protocol. Thank you.
That's awesome. Thank you, Asma. We're moving into Saba's portion of the presentation now.
Okay, so now I'm going to tell you about the media that I chose and its properties, basically, that I checked. Next slide, please. So as you see, like, I use like T cells, and that's the media that that's a cell that, I usually work with. And I wanted to increase the cytotoxicity and basically function of CAR T cells. And then also I wanted to maintain the viability and activity of the T cells. And there are like several components that I will go through rationale that why I picked that. And, in the next slide. So if you go next slide, please. So just to recap from the last webinar, I mentioned that there are multiple factors that impact the efficacy of CAR T cells. It could be CAR design or the factors in tumor microenvironment, but one of the factors that we have kind of a full control on it is the quality of CAR T cells. And obviously, this is influenced by the fuel that we provide for these CAR T cells. So next slide, please. Here, this is my experimental outline. I used 2 different processes. One is the standard process that we use in the clinical sector. And in this process, we get the T cells and we activate them with Dynabeads. And after a day, we add lentivirus vector, and then we expand them for several days. And at the end, we can check their functional properties. And the second process is my personal approach that I developed at Penn, and it's the use of non-activated CAR T cells. So in this way, we actually do not activate CAR T cells. We keep the T cells quiescent. We just get the T cells, and then we add lentivirus vector. And then after a couple of days, we can check their gene transduction and then check their functional properties. Next slide, please. So these are basically the components that I picked. I picked IL-7 and IL-15 cytokine because they are cytokines that are permissive for gene transfer for non-activated T cells. When we keep the quiescent T cells and we want to have gene transduction, we need to have this cytokine. Also, next, I wanted to see the impact of lowering the sugar level in the media, because several studies show that the sugar level is not optimal. So that's like another thing that I wanted to see and see the impact of it in the CAR T cell function. Also, as I explained before, I wanted to check physiologics because, we also showed that, that impacts CAR T cell function in mouse model of neuroblastoma. Another component that, we're very interested in right now is arginine. Again, there are studies that they show that T cells that are grown in media, including arginine, it has, they show anti-tumor, higher anti-tumor function in mouse model of melanoma. So that's another component that I was interested in to see, if I add that to my media, can I change any impact of, CAR T cell function? And also the last component that I was interested in was carnosine. Carnosine is actually physiologics contain carnosine, and that's that control the pH of the media, which is important for gene transduction and CAR T cell function. Next slide, please. So one of the important determinant of the current approach that we use in the clinical sector is the proliferation of the T cells. So in series of preclinical assay, we always interested to look at the proliferation of CAR T cells and their viability. So we do this rigorously at a regular interval with flow cytometry. And we monitor also the cell size, which is an important determinant of the activity of the T cells. So as you see in the graph on the left, which we show the population doubling, that over days, my control media, which is red, by day 10 it plateaued, but cells that are grown in the customized media that I picked, they're still growing, and this is this shows that basically that they are they have higher function. Also, we can see the cell size, which we see that by the end of the cell size is higher at the end of by day 10, the cells size is higher in the custom that cells are expanded in customized media compared to the control media, which suggests that the activity is high. The activity of these cells are higher. Next slide, please. Another parameter that I wanted to look is the transduction efficiency of lentivirus using this media in my two different processes, either cells that we activate or the non-activated T cells. So on the top part, we see in the activated T cells, we see we use a fluorescent protein MRFP, and then we see similar MRFP expression level in the activated CAR T cell context. Both of them are almost a similar level. On the bottom graph in the non-activated CAR T cells, not CAR T cells, sorry, these are only T cells, and then there are the lentiviruses MRFP. We actually noticed that the transduction efficiency in the customized media is lower than the control media. So this is important, and this is great result for me, because it's important to notice that the factors that I picked, they're not optimized for this process, this specific process. So I have to explore more factors to see how can I boost the gene transduction in the non-activated T cells. Next slide, please. Another important function that I wanted to check was the metabolic activity of these cells, because there is the very high interest in the field about the metabolism, and we know that the metabolism is linked to differentiation. And as I explained in the previous webinar, that we always wanted to have less differentiated T cells. That's why actually the second process of non-activated came in, because we wanted to just eliminate the activation which trigger robust differentiation. So next slide, please. So here in the graph, we check the oxidative state of cells that are expanded in the control media or in the custom media. And as you see, the oxygen consumption rate of cells that are expanded in control media is almost half of the custom media. So it tells us that, the custom media actually increase the oxygen consumption rate of the cells, which is a great function, and it actually, it tells us that they would rigorous, let's say, environment, they would act a lot better and they have like higher functional ability. So now, knowing this, I would really like to explore other candidates that I can manipulate, for example, in the media to see, like, if I can optimize my lentivirus gene delivery and maintain my functional competence. So one of the things that, I was thinking that, maybe for that 24 hours that I do use for the gene delivery, we can use the control media, and then after that we can switch it to the custom media, so that in this case, like, we can have like a higher gene transduction, and also, like, maintain the functional competence of the cells that are grown in the customized media. And overall, the whole process was very seamless and very easy to apply for future studies. So that's also my plan, to have, like, different components, and see if I can optimize my media further. That's it. Thanks.
All right, I think we're moving over to Lori now. Great to see those really interesting results. It's awesome to see how the small changes can really affect the outcome, how that spurs ideas. Oh, Lori, I think you're on mute.
Always, sorry. You said exactly what I was going to start off with. Those are really interesting results, and I want to thank you guys for having me participate in this webinar, because I've learned a ton. Those of you guys that know me out there know that I have more of a process focus than an assay focus. So I really enjoyed Saba's and Asma's data. However, Cognate is a CMO, so most of our processes that we're contracted to perform are somebody else's. So I selected an internal project for sharing with you guys. Part of the process group within our technology development job is to provide material for the assays group. So that's kind of where I went. Cognate is a part of a Charles River company, so we actually can produce cells for a lot of different assay groups. When they need cells for development or fit for purpose evaluations. Specifically for this, we chose to grow some Tregs, which were going to be used for a flow assay. So proliferation of viable T cells with the correct surface markers was our primary objective. And the next slide summarizes the experiment. So basically what we did was we took just a Treg profile protocol from Miltani and Fisher, and kind of combined the two. And so the experimental design was just using those kits for the Tregs to do the expansion. And specifically, I was looking to increase proliferation, so I'd have enough cells to test, while maintaining, as I said, the viability and the CD4, CD25, and the FACS3P3 FACP3 phenotype. The bar is a bit low since I was using that type of an assay. If I were to use a functional assay, I would have been way more concerned. I would have had some other more interesting CQs to put in here. So my experiment did, as expected, result in some pretty CD4, 25, FACS P3 cells. And the next slide shows that the viability, was decent, and the morphology was what I wanted. Not shockingly, the cell proliferation was still a little bit better in the proprietary media. I'm sorry. So proliferation was, like I said, a low bar. That was just, and so my next steps, I think, are really exciting, because that is to look at the medias to use different things. Now, I get that trying to make the cells do different things so that they work different in medias isn't really everybody's gig. And that is, it's not even my gig all the time, but the ability to tweak the media to engineer the products that will demonstrate varying functionality is such a powerful tool to keep in your, bag of development tools, that it is worth, it just demonstrates how powerful this can be, and that anybody can do it. Like I said, this isn't what we normally do, but it was fairly easy, and it was fairly quickly. And so my next steps are going to be, first of all, to re-stimulate the ones that I have and put them in for an MLR comparison, to test them a little bit to see if there was any difference in the functionality, but also then to use this MB process to produce other cells for development, both the process aspects of it, as well as continuing to make cells for different types of assays.
All right, thank you so much, Lori. And that's science, right? Sometimes we get the results we anticipate, and it spurs a lot of ideas, and really allows us to to tweak things to try to get that desired outcome, as you were stating. And the perfect example there, knowing, and that transparency of knowing what's in those custom media is also quite an important aspect of every study, and the ability to be able to tweak that to get your desired CQA. So thank you all for working with us and doing this little online experiment with us. It's been a lot of fun. I hope you guys had a lot of fun, and we look forward to working with you guys more going forward. I think if we move to the next one, Rachel, just a quick recap, not really a recap, but just a list of the other learning resources we have out there that have introduced MBLux, MB Air to you guys, our customers, and the audience out there. You'll find the link at the bottom, which will take you to those resources, and you can watch them, view them at any time. Or if you have any questions, also have emails at the end of this. Please feel free to reach out to me directly, or to our info at nucleusbiologics.com. If you can advance to the next slide, Rachel. Just want to leave you guys with a few final thoughts about Nucleus and what we stand for, and why, again, we're so excited about custom media. Why choose us? We believe in customization. We've been able to improve ordering efficiency, expedite times. We believe in the customization and reducing waste and only ordering what you need, reducing labor and time. Individual ownership is huge. Knowing what's in that media, as each one of our scientists has pointed out, is really, really beneficial in being able to tweak that media in order to achieve your desired outcome. Consistency, streamlined QC validation, everything goes into the media here. Essentially, you're ready to use it once you uncap it. Scalability, we are there with you from, like I said earlier, from the bench to the bioreactor, from 2 liters to 2,000. Transparency, no hidden ingredients in our products whatsoever. It belongs to you. They're your choices. We're certainly there to help you if you need it, but certainly your choice as to what goes in that media. And cost effectiveness, right? So certainly the smaller batches come with a bit of a price premium, but we're there to scale with you. And at scale, we can certainly help you realize cost efficiency and save you even millions of dollars as you proceed down that pathway. And finally, AI, or technology driven. We're using cutting edge technologies, cutting edge proprietary algorithms in order to really drive and expedite the formulation of your media. And we're trying to do this in a very automated fashion. And we look forward to working with all of you out there to even make it better as we progress, through the years. So hopefully you guys will reach out to us and let us know how we can help you kind of achieve your goals and reach your CQAs. So I think at this point, we would probably move into the Q&A section. I'll turn it back over to Rachel. I think she's going to moderate that for us. And again, appreciate everyone for joining. Please put those questions in the chat box or the QA box.
Yeah, absolutely. Thank you, everyone, for sharing. That was very enlightening. We have a couple questions here, but please do send more through the Q&A and the chat boxes, and we'll get those addressed. But Saba, we had a question for you about what your control media was. You're on mute. Saba, you're on mute.
Sorry.
There we go. No, there we go.
So the base of my media was basically RPMI.
Okay.
Is that like, if the base of the media was, yeah.
Great. And then we also had a question relating to stability and, why we don't do stability for the media. And I think I'm going to ask Lori to take this one. If you can speak a little bit to stability.
So a lot of you, from the process point of view, as a lot of you would like that to be stability tested as you come in. How long do I have before I have to use it? But the reality is, as you go towards your GMP and commercial processing, that you are going to have to look at that individually yourself, regardless. And looking at it with a consistent media that is already prepared is way easier than looking at it where you're adding the components and you have to look at it, after you add which components is it stable, when you're thawing it out. I mean, you already have a lot on your plate as you go through the process qualification when it comes to your media. So you're going to have to do it. And I'm here to tell you that it's way easier to qualify a media and to determine the stability on a media if that media is consistently made and you don't have operators that are making it, and you don't have to do that check. So the media that comes in is already made, it's already tested, you already know what it is, and so your stability results are going to have less variables associated with them.
Thank you. Any other thoughts from the panelists on that? Okay. Any other questions or comments? Please send those through the chat in the Q&A, and we'll definitely address those. We don't have any at the moment, but I'll wait a minute and ask if the panelists, after all that was shared, have any final thoughts or nuggets to leave us all with. Okay. Well,
I mean, I really enjoyed this process, and I certainly, appreciate everyone for going on this journey with us. And I love seeing the results, and I think it's fantastic to know that, this really shines a light on ways in which you can make your media better and make your formulations more effective. So. Yeah,
I'm excited to see and hear how all you guys continue to use NVAir to refine your media and iterate. And we may not have a formal webinar to present it, or maybe we will, but I'm excited to continue hearing all of your data. We had a question, actually just had a question come in. How long would it take for a custom formulation to become GMP available from project initiation?
Rachel, you may be able to speak to the process there a little bit, or Asma.
I think it's mostly Rachel, right? Because it's manufacturing question. So yeah, I'm more in R&D.
Yeah, and that's an interesting one. We can move quickly through the process. So as you saw, everyone started in their small lot, but then to move to full GMP, it should take about 12 weeks to get through that process with Nucleus.
Yeah, as you guys saw, the turnaround time for the smaller lots was about 2 weeks. So, certainly, but it does vary. CGMP comes with certainly a level of document control and complexity that does extend the time frame, right, which is anticipated.
Yeah, but those are the projects that we love working on. So if you're there, please contact us, and we'll get there with you.
So, and it would probably flow, that 12 weeks would probably flow with what the site has to do, the manufacturing site has to do. Because if they're getting ready to, to gear that up, they have to do their vendor qualifications and some, fit for purpose points too, so that it's GMP. There's a ton of paperwork that goes along with it.
Absolutely.
Yeah, absolutely. Well, we'll still accept questions, but since I did just say contact us if that's where you are in your stage, I want to make sure you know how to do that. So we have our contact information here as well. But we will stick around if there's any more questions or answers that come through. We'll love to answer them. But if not, we want to thank everyone, panelists and attendees included, for their time today. I know everyone's quite busy, and we love having you here. And thank you to the panelists for all the hard work and sharing of your work.
Thank you.
Absolutely.
All right. I hope everyone has a fantastic rest of your day.
Thank you.
Thank you.
Take care.
Bye-bye.